Hi everybody, today I want to speak about biochemistry. I start with the amino acid and the proteins, and then I will continue with the other parts. Okay, the first part I start with the amino acid. In the biochemistry, we have several types of the amino acid. Amino acids, okay, has a carboxylic group and amine group. Okay? And we have several types of the amino acids. Okay? Depend the position of the amine, because when we have a amino acid, the priority of the carboxylic acid is higher than amine group. Okay? Then if we have a, for example, this carboxylic acid, if the amine group in the alpha carbon, we call it alpha amino acid. Or if amine group in the beta carbon is the beta amino acid. Okay? And if the amine group in the gamma position, we call gamma amino acid. Okay? Mainly for the peptide formation, we have alpha amino acid, but in the other in the body we can find also beta amino acid, and also in the pharmaceutical part we can also have a gamma amino acid. Mainly for the peptide we use the alpha amino acid. Alpha amino acid, okay, has one carboxylic acid group, one amine group in the alpha position, and one hydrogen, and one R group. Okay? We have 20 types of amino acid, alpha amino acid, which the R groups can be different. Okay? And as you see here, if the R is equal hydrogen, the amino acid is the glycine. It is the achiral. The only amino acid which is achiral, okay, it is the glycine, which R is hydrogen. But other amino acids all are the chiral. They are optical active. It's very important, and we have to care about this, because the amine group and the carboxylic group and the hydrogen group, all three with a different R group, is chiral. And they are, as you see here, the amine is the primary, all, but then we can see is others chiral. And all of amino acids are primary amine, except proline. Proline is the secondary amine. It's very important, as you see here. The position, as you see here, the position of amine is important. If we have a just bind for the one carbon, okay, as you see here, just bind for the one carbon, and it's alpha. But in the proline, we have a different. We have a two carbon bind for the amine group. Okay, amino acid, okay, they are amphoteric molecule. Okay, they can react with the acid or base. Okay, they can react with both the acid and the base. Okay, it's important. Okay, for example, if we have amino acid in the solution, if you react with acid in the solution, in this case, the amine group can accept proton and become NH3 positive. Okay? the net charge of this molecule is a positive. Net charge is a positive one or positive because the amine group accept proton, and it becomes net charge is a positive. But if we react with a base, for example, sodium hydroxide. In this case, the carboxylic acid group lost proton and become carboxylate, and the net charge is equal -1. Okay? As you see here, we can say if we increase the pH, the net charge will be negative, and if we decrease the pH, the net charge will be positive. But there is an important thing, the effect of the R group, because the R group can affect in the which pH will become net charge positive or negative. Okay? For calculation of this case, we need to know information about isoelectric point or pi. Every amino acid has an isoelectric point, okay? Because pi is equal, because this one, it has a pKa1, and the amine group has a pKa2, and normally the pKa1 plus pKa2 divide 2, we can calculate the pi isoelectric point. Okay? Also pKa1 and the pKa2 can be affected by the R group. Okay? Okay, directly we don't see the R effect in the formula, but because R group can affect for the acidity of this carboxylic acid or basicity of this amine group, in this case, the isoelectric point can be changed. It's very important because in the organic chemistry we studied, for example, if we increase the number of the carbon in the carboxylic acid, the acidity decrease. And if we increase the amine group, the basicity increase. And this can affect the isoelectric point. It is the situation, the net charge is the 0, and we have a Zwitter form. In the Zwitter form, the amine group accept proton, and the carboxylic acid lost the proton, but the net charge is 0. Then in this case we have a both positive and a negative. It is when the solution, the amino acid, if the pH equal the isoelectric point. If we at the pH equal to pi isoelectric point, in this case we have a Zwitter form, Zwitter ion. Okay. Now we understand, it's very important, we know for every amino acid, when pi equal pH, okay, and after this point, we increase the pH, we can have a net charge negative. If we decrease pH, we can have a net charge positive. Okay? And if we have a peptide, for example, we will study later, the isoelectric point of the peptide is equal the average of the isoelectric point of all of the amino acids. Okay? In this case, we can say this peptide is more acidic or basic. For example, if we have a carboxylic acid group in R, for example, glutamic acid and aspartic acid, in this case, the isoelectric point will be more acidic and will be less than 6 and will be around 2 or 3. Or for example, if we have amine group in the R group, like lysine, histidine, the pH isoelectric point will be in higher pH, for example, around 8, 9. And it is very important. We know this case, how we can calculate isoelectric point. Now I continue with the classification of amino acids As you see, as you know, the classification of amino acid depends for the R group. Okay? And because of the R group, we can classify the isoelectric point. And we can calculate how is the property of our amino acid. The first group is the aliphatic nonpolar amino acids. Okay, the first group is the aliphatic nonpolar amino acid. Okay, for example, I write here aliphatic nonpolar. Okay, the R group are nonpolar compound. Okay, for example, I start from here. Okay, if we have R group, hydrogen, it is a glycine. Glycine, it is a nonpolar amino acid. Okay, and it is the only amino acid which is not chiral. Okay, and isoelectric point around will be 6, because as you know, the isoelectric point, because the pKa of the carboxylic acid, it is around 2.3 or 2.4, and for the amine is 9.6 or 7, and the average will be around 6. This is the glycine. Okay, the second amino acid from the aliphatic nonpolar is the alanine. Alanine, the R group, is the methyl group. As you see here from the chemistry, the acidity of the glycine is more than alanine because this R group decreases acidity. Okay, and but alanine is the chiral compound because the 4 group different in the alpha carbon. And it's very easy. As you see here, it is a second group. But if we have, instead of the R group, isopropyl, for example, if we have an isopropyl here, isopropyl, the amino acid will be valine. In the isopropyl, the structural amino acid will be valine. And this is very important because the valine structure, the number of the carbon increase and the acidity of the valine is less than alanine and glycine. In this, from the history, the glycine has the highest acidity. And the next one from the aliphatic nonpolar is the leucine. If we add the isobutyl instead of R group, it is the leucine. For example, if we have a isobutyl group, isobutyl, CH, isobutyl, it is a leucine. In the leucine, the R group is the isobutyl. As you see here, it is also a nonpolar aliphatic group, and it is very important. The other one is the isoleucine. In the isoleucine, instead of the isobutyl, we have a secbutyl. For example, if we have, for example, the same group, but we change here just for the secbutyl, it will be isoleucine. As you see here in the isoleucine, the secbutyl is instead of the isobutyl. It is very important. Okay? These are nonpolar. Okay? But in the nonpolar aliphatic, normally we have a carbon and hydrogen. But also we have a nonpolar aromatic or cyclic nonpolar. In the nonpolar cyclic, we can have benzene ring also in this group, but there is an exception in the nonpolar aliphatic is the methionine because it has a sulfur, but sulfur usually is the polar in the compound because it is between the two carbon group. In this case, leucine, methionine will be also aliphatic nonpolar. Okay, this it is the exception for the sulfur group because in the we will see the polar in cysteine also. It has a sulfur because it's one side is open. In this case, it will be polar. Okay. In this case, as you see here, if we have a sulfur group between two carbon. For example, I have a CH2, and we have a SCH2, as you see here, CH3. Okay. Pair electrons, it has the two pair electrons, okay? This air S is the between two carbons, okay? It is a nonpolar. If with these groups, we change this group for the hydrogen will be polar. But it's an exception because this sulfur hasn't polar effect, but or it has a very, very small polarity, but it's almost nothing, okay? It is the other one. It is, and we go. Also, we have before we finish aliphatic. Okay, there is a proline which I spoke about it. It is a secondary amine group. It's a secondary amino acid. It has a, as you see here, this amine group bind for two carbon. One is this one, the alpha carbon. The second one bind to the other CH2, as you see here, and the CH2 and the CH2. Okay, you see the R group rotate and again bind to the amine group. It is a proline. It is also aliphatic amino acid, which is nonpolar. Okay, as you see here, I drew all of them. The first group is the glycine, alanine, valine, leucine, and isoleucine. All just has the carbon and hydrogen. They are aliphatic nonpolar. There is this exception with the sulfur group is methionine, which is S group is between two carbon group as the nonpolar, and there is a secondary amino acid which is the proline, and this amino acid also is nonpolar aliphatic. Now we go to the second group. The second group is the nonpolar aromatic, or non-aliphatic, or aromatic. Aromatic or cyclic nonpolar amino acid. They are aromatic or cyclic. It's very easy, important, and very nice. The first one, which always it is very easier one, which when we have a benzene ring because it's aromatic. Okay, as you know in the chemistry, you know this structure called benzene, and if we have CH3 is the toluene. Okay, if I bind instead of toluene, I bind this group instead of this hydrogen as the alpha group. In this case, okay, this structure changed for this structure. Okay, it has a hydrogen, CH2, and one benzene group. As you see here, it is phenylalanine. This amino acid is phenylalanine. It's very easy. It has a benzene group which binds to the CH2, and it is a cyclic or aromatic nonpolar side chain. And in the chemistry, maybe you remember or know, if we have this structure with a pyrrole bind to the benzene, we call indole. If we bind indole group to the amino acid, in this case, this will be, as you see here, and it's very, very easy. As you see here, I bind from here, NH2 and H. As you see here, it's one of the very important amino acids. It's the tryptophan. It used in the body. We will see in the next lecture for synthesis of the serotonin, which is very important. It is one of the neurotransmitter in the CNS, which helps for the happiness and prevention of the depression and anxiety. Is one of the important groups which is we studied out here. These are non-polar side chain, which can be divided to aliphatic, non-polar, and aromatic non-polar side chain, and it is very easy, and almost the isoelectric point of these groups around the 6 okay, can be different because of different R groups. As you know, when the non-polar R group is the bigger and the number of the carbon is higher, in this case the amino acid will be more basic in the pH compared to the less number of the carbon. Okay, and I will go now to continue to the polar amino acids, and I stop here and I continue in the next video, and we will see in the next video about the continue these groups. Thank you.